Good afternoon. I will be really brief. I have just few slides, and I will focus on what we are doing and how you can access us. So, what is our main instrumentation and services, and our main topic? Now it's genomics, and most important genomic tool today is massively parallel sequencing. So we are focused on massive parallel sequences. We have the instrumentation, we have the knowledge, and we have all the accessory instrumentation. And thanks to bioinformatics, we have also the analysis platform. So what we have, massively parallel sequences. We have another machines like robotics, UPCR, or single-cell machines. And our main services are library preparation and sequencing. Easy. And how you can access us or use us. We offer all access modes. It means full service or assisted usage or self-service. It depends on the instrumentation and your skills. So if you are new in the field, the best option is to ask for full service. If you are experienced, at least a bit, you can prepare libraries yourself and just bring libraries for sequencing, or you can do sample QC on tape station or fragment analyzer yourself. And for some standard machines like qPCR, we are offering no assistance because you should know how to use qPCR and how to pipette PCR. What is very important for our users is fees. We always ask for 100% consumables because our consumables are expensive and we cannot pay them from any project. Sometimes there are open access, open course projects like Alliance for Life projects. So you can ask for sponsoring for consumables, but for normal users, they always have to pay the consumables. And for the instrument time and our work, it depends on from which organization you are. So people from SATEC will pay less, something like 15%, and commercial companies will pay 100%. And straightforward and clear, and it's in our prices. What is new? Years 2022-23 are very important for the whole field of massive parallel sequencing, because this summer the last patent that was held by Illumina was not invalidated but expired after 2 years. So since mid of the last year, there is much more companies on the market that start offering really competitive systems. And happily, we had also investment window from different projects. So now we expect until the end of the year these 3 new instruments. NovaSeq is already here. It's a Illumina-based system for high throughput sequencing at a affordable price. Now, unfortunately, there are new machines that offer lower price. Then we expect Element Bio, which is one of the most important systems on the market now because they changed the way how the sequencing is done by the sequencing chemistry, which is different. And we also have now for limited time, maybe it will be prolonged, this MGI DNBC G400. It's another massive parallel platform. It's benchtop and it's of Chinese origin, but Chinese get the technology by buying the Californian company that was very competitive to Illumina 10 years ago. So they are again very competitive, and especially for the price. Unfortunately, some of these new systems are hosted for XLS projects, which means they are available only for people that are part of these projects, unfortunately. But we are at least learning how to use them, and after NPO will finish, it should be open to everyone. Now, very, very important slide. We have it every time. Acknowledgements, please acknowledge us, and definitely we prefer the one with the project number. And if you are not sure, you will see this. It's our webpage. Please, if you, most of the information are there, so visit our webpage. And if you don't find information there, ask us. We also are trying to use ticketing system in the booking, but so I would like to encourage you to use this booking system to register and ask and register your requests. Again, our webpage. And I think the last slide is about our data policy. It's the same as the last year. Still, we guarantee for sequencing, we guarantee 6 months only. That's all. And for small machines, we guarantee nothing. That's all. And thank you for your attention.
You are going to face a lot of questions during the next 10 minutes.
Really?
You are very fast.
There will be second coffee break.
You talked about short read sequences. What are they? What does it mean, short?
Short and long. Long reads means kilobases, tens of kilobases, hundreds of kilobases in a single molecule. And short current limit is 2 times 300. It means from the fragment that could be up to 1 kb, you are reading 300 bases from one end and 300 from second end. So it's a very different technology, even for the reading, because for long reads, the sequences are called those single molecule sequences. They really take single DNA molecule and read the sequence. Short read sequences, most of them are using somehow amplified DNA to have nice signal. Most of them use fluorescent labeling of bases. So it's fluorescently based, it's microscopy. Again, some type of microscopy. Long read sequences are specific. They could be very cheap. Even you can get the sequencer for free, not a problem. But their error rate, because they are reading single molecules, so the error rate is a bit higher. But it can be improved with other ways. What is nice on long reads, usually they can read nascent DNA, so no modifications of the sequence, so you can read even epigenetic modifications. And for ONT, you can even read nascent RNA molecules with all the modifications, but we never tried.
But do you have this long read sequences as well?
Yes, as I said, it's very cheap. So you can get it for free, you just need a computer. And we have also the larger one, and the sequencer itself is 10,000 euros. So it's a cheap machine compared to others. And this 10,000 euros includes chemistry for 9,000. So the sequencer itself is for 1,000 euros if you want it. And it can sequence 2 whole human genomes within 2 days. So again, the capacity is not small. The price for the genome is around 1,000 euros. So it's even not very expensive for the analysis. Okay.
Let me ask, can you tell us something about this XLS project that prevents us from using your machines?
You don't know it? Lucky. These are the centers of excellence. I don't know. It's National Sustainability Program. No. National Reform Program.
Oh.
Okay, I don't know the English name. I know the Czech name, and these are these big institutes. I think biology and cryo-EM knows a lot about the projects because they are. It's biology, microbiome biology project, cardiology, oncology, neurology, and social medicine, or somehow it's called. So these are big projects, but with very complicated rules for funding.
So does it mean there are like specific people involved in the project that are using the machines, or is there some other type of usage?
The machines can be used only for the project. So it means it's only for the people that
Are involved.
Which CETEC is involved, I think, in almost all of the projects, or Masaryk University is in all the projects, and CETEC itself is, I think, in 3 of them. But I'm not sure. I think it's still not even possible to cross-project usage. So virology cannot use oncology, and oncology cannot use virology machines. And also, value VAT is not allowed, so you have to pay the VAT. Another complicated rules. But the projects will end in 2025.
Michal mentioned some interesting user projects implemented in the facility. Can you also maybe mention one or two?
It's our users project, so but I think Vojta, you will talk about it because for us it's very important that we can offer complex analysis. And our nice projects that we have is sometimes it's sequencing of some rare species. You can sequence because sequencing every living organism has some nucleic acid, and we can sequence it. So we saw really small projects like sequencing of dead dolphin. It was beluga from Black Sea. They found it on the beach. They took it somehow to Czech Republic. And it's not recent project. It's, I think 6 years. It was not even a project, but they were curious they were looking for canine distemper virus. It's in Czech, it's psinka. This virus can affect also sea mammals like dolphins and sea lions. So they asked to try to sequence RNA or cDNA from the sample to whether there is a RNA of the virus. And I think from the reads there was one read that was from canine distemper virus. And we have also many, like our customer for years. They are doing parasitology, and they are analyzing for years samples from I don't know whether mountains or the second species of gorillas, and comparing with chimpanzee, bonobos, and humans from Africa, and trying to see the transmission between species, transmission of the species. These are quite nice projects, long-term projects. Vojta will talk about another one, hopefully. Yeah, even cross projects that try and combine some medical imaging with genomics, or some nice projects trying to combine some projects trying to combine proteomics with genomics and others.
Do you have any projects from archaeology?
Let's say Mendel. Well, yes, we have our clients are also from department one of the departments of molecular archaeology at Masaryk University. But special project was sequencing of Mendel genome. It was done here, but I like more these standard projects that are looking for hundreds of years old remnants, and they are trying to find which population it was from the mitochondrial DNA. They are also looking at the pathogens, and so they are also sometimes looking for bacterial species that are found there. So yes, we have archaeology, and archaeology is very, very specific because the material is very difficult to handle and is very sensitive to contamination. And the quality of the data is not as good as we expect for medical procedures.
Last question, sorry. During our yesterday's call, I've learned about the new method, optical mapping. I've for the first time heard this method. I guess the instrumentation, there's no instrumentation here in our course.
No, and what we want to also prove in the project that long read sequencing is as same good as optical genome mapping. So then that is one of the goals. So we are looking for optical genome mapping. It's a very specific method to analyze long DNA molecules by positioning fluorescent marks. So it's only for resequencing for well-known genomes. And it's very specific, and it's offered only by one company. And we also think that optical genome mapping is nice, but it can be replaced by long read sequencing. Because it could provide very similar information and even better information at the same price.
Yeah, some on-chip method.
Yeah, this optical genome mapping, there are very tiny long and tiny channels, and the DNA is stretched. So there is one molecule of DNA per channel, and it's so you can see it in linear form, and you can place different markers repeatedly and map the position of the markers and then assemble the information back. It's method, I think it's 6 years old at least, and now it's popular because it offers information on a really long scale, like hundreds of kilobases, but still long read sequencing can provide very similar information.